Okay, so yeah,
This is the water output.
huh?
This is the water output.
Oh, that's fine. Yeah, I already put it. That's fine. We can do anaerobic. It's fine. Don't bother. Yeah, like I will. So there are two review lectures. One is anaerobic respiration in hydrocarbons, and the other one is phototrophy and fermentation. So it doesn't matter which one we do first. This one has 24 slides. Again, I will just go through them. Please don't think that you don't have to touch anything else, like only review lectures. Read everything, but you will get good mark even if you only go through these lectures. Like the reason why I make this review is so you can have easier time studying. Okay? And this will be even more important for midterm. Midterm, I will probably use only these reviews because it will be a lot. So I don't want to bother too much. Anyway, so we are here to review anaerobic respiration and hydrocarbon metabolism. Why did I press Brittany? Come here. I have no idea what did I do. Sorry. I'm not going to touch anything. I knew it. Every time when I touch it, it will be. My husband says that Bill Gates should hire me. If I touch it and I don't break it, that means it works. See? Thank you. I'm not going to touch it. Okay, I'm not. God, demonic invention. Okay, so speak about something that is not demonic invention. Meaning, metabolic diversity, anaerobic respiration, and hydrocarbon. Brittany, these things hates me. Technology doesn't love me, and I don't love technology. So it's kind of neutral. So when we talked about anaerobic respiration, what that means respiring something other than oxygen. Cells are breaking down sugars, generate energy in the absence of oxygen, and we talked about power of power, where oxygen is on the bottom, which means it has a very high redox, very positive redox potential, while you have other terminal electron acceptors that are listed here, and all of them I want you to know. NO3 minus, NO2 minus, nitrate, nitrite, sulfate, carbon dioxide, proton. Proton will be the most interesting one that I really like to ask because I really love the name of the organism called Spirococcus furiosus. I really like it. I don't know why, but it's so interesting organism. In terms of, so again, tower of power, if it is on the top, it is not good terminal electron acceptor, but it is good electron donor, which means it has highly negative redox potential. If it is on the bottom, that means it's very good for the energy production. It's the best possible terminal electron acceptor. It means it has highly positive redox potential. Again, think about that. Electron is negative. Highly positive redox potential means very good. Oxygen is the best. Is the sulfur, which is mid one, but oxygen and nitrate are the best. When we think from evolutionary perspective, earlier it was overloaded with hydrogen sulfide, and iron 2+, and microbes evolved mechanisms to extract electrons mostly from these three components because they are highly reduced. Again, know that they couldn't respire oxygen because it wasn't available. So all these 5 terminal electron acceptors you have to know. When we talk about and the terminal electron acceptor, this is called nitrate respiration. And again, this is where the confusion came next time. We will talk about denitrification and nitrate respiration. They're not two same processes, but for now, like since we didn't cover denitrification, only know that NO3 minus get reduced to NO2 minus, and there are enzyme reductases that are involved in that. If there is proton motive force involved, that means it's meant to do energy generation, but very often that doesn't happen. Again, denitrification, it's a process that does not involve energy production. There is no proton motive force. And it's bad for agriculture, but it is beneficial for sewage treatment because it's helping reduce NO3 minus nitrate waste in the process. Nitrite is another electron acceptor that is usually done by Anamox, and I want you to know to recognize this profile, the anamoxidants and methylomirabilis oxifera, because they are two organisms that are respiring nitrite as terminal electron acceptor. Anamox, I will really insist that you ask multiple questions about what is energy source for them. So it's ammonium, and that one gets oxidized, and they respire nitrite, NO2 minus, which gets reduced. What they produce is hydrazine and nitrogen. Hydrazine is very toxic. And again, anamoxisms, I call them pseudo-organelles, but Brittany has a paper that says that they're called organelles. So you're free to call them as you want. I'm not going to ask that because, again, I'm not expert in the field of anamox. So I can call it Susan if I want. People who are experts in it, they call it organelle. So it's only my opinion. What you need to know is that anamoxidants are surrounded by special types of lipids that are called bladderan lipids, and they have very specific bond that is non-ether ester. So I think you are right, Brittany. That's where they actually remind to this one that is that exists in thermomicrobium. Oxifera is Oxifera will be enough. I will never know how to pronounce full name. They use methane as energy source, analog to ammonium in anabox, but they also respire nitrite and they produce nitrogen and oxygen. Think about that. Nitrogen and hydrogen. These guys produce oxygen, but oxygen is only used to activate methane oxidation, which means that makes these organisms anaerobic because they do not respire oxygen and they belong to this NC10 phylum. Sulfate is mid ground in terms of energy. So because a mid ground labeled here, that means in order to connect them with some energy donor, some electron donor, it has to be on the top, which means it can be only connected either to hydrogen or to some organic source, because you have to have that enough big difference. So when they oxidize hydrogen or organic source, they reduce sulfate, which means they produce sulfide, H2S, which is the reason why we call them sulfidogens. Another name for them is SRB, or sulfate reducing bacteria. Again, SRB, like Serbia, my home country. So that's how I memorize. I will never forget it. The characteristics of this sulfate reducing bacteria is that they use either organic or hydrogen, they respire sulfate, and they make this hydrogen sulfide that will be delivered to anoxygenic phototrophs because they need that H2S to run photosynthesis, and they will be in competition with methanol because they both use hydrogen as fuel source. What is interesting about sulfate reducing bacteria is that they are actually causing corrosions on the bridges. If you think about metal surface like a bridge construction, so there will be those microbes on the surface that will consume all available oxygen, and then they will make that area oxygen depleted or anaerobic. Once when it's anaerobic, that will be a special place for SRB because they are anaerobic. So they will reduce sulfate and generate sulfide, and sulfide will react with metals that are used to make bridges, and that metal sulfide will create corrosions. So that's why very often bridge constructions are coated to prevent microbial biofilm formations. Symbiosis between SRBs and ANME is very important to understand. ANME are anaerobic methane oxidizing archaea that are consuming methane, and they reduce, so they live in consortium with sulfate reducing bacteria. So ANME use methane as a carbon source and they respire sulfate. Sulfur reducing bacteria will use organics as energy source and they also respire sulfate. So since this is symbiosis, SRB creates energy like NADH when respires sulfate, and then ANME is using that NADH that is made by SRB. So I understand where it's like ANME has connection. Like ANME has benefits. What kind of benefits has SRB from that? I'm not really sure, but it must be something. Probably ANME is making some organic compound that is delivered to SRB, but I did not tell you about that. One of the old ancient metabolism is acetogenesis and methanogenesis. Both of them use hydrogen as energy source, and both of them respire or fix carbon dioxide. So I put under like quotation because it's not really the same process. What characterizes acetogenesis is the ability to have CO dehydrogenase and hydrogenase. And most important, they use hydrogen as energy source and they fix carbon dioxide with Wood-Langdale pathway. Methanogenesis, similar product. So acetogenesis produces acetate as a metabolic product. Methanogenesis produces methane. Methanogens compete with sulfidogens for hydrogen source. That's important. Then we talked about metal reduction, ferric ion and manganic ion, 3+ and 4+. Again, it has to be in that structure because otherwise cannot be respired. We talked about Shevanella and Geobacter as important iron reducers, and we will talk more about them later on. They use as energy source, they use hydrogen, but they respire metals. And the Shevanella and Geobacter are known by production of nanowires, which are literally structures made out of cytochrome that are doing like a protrusion through membrane, and then they attach to the solid surface, which is how they respire. Again, we will talk more about that in the time. And this one that I really, really like is Archaea that respires proton called Pherococcus furiosus. Lives at 100 degrees, very hyperthermophilic. And again, when we talked about different electron acceptors, desfupomaculatum performs arsenate and sulfate reduction. This is all what I want you to know, that the reduction generates mineral orpiment. So arsenate respiration is arsenate is toxic, but these guys can actually use it as energy as terminal electron acceptor. In terms of hydrocarbons, one common feature is that they have to be activated with oxygen to be used as an energy source. And enzymes that are performing this activation are called oxygenases. It can be mono or deoxygenases. Monooxygenases will incorporate only one oxygen and the other one they will reduce to water, while when you have deoxygenate, it will incorporate both oxygen atoms into the molecules. Again, famous deoxygenases are in Beck's degradation. Methane degradation is done through monooxygenase. We also mentioned hydrocarbons, aromatic or aliphatic as a carbon source. So alkanes, alkenes, alkenes, and the product of oxygenation is fatty acid. If hydrocarbon is aromatic, the product of oxygenation is catechol. That's a big difference between them. When we talk about aliphatic hydrocarbons, we talk a lot about methane oxidizing bacteria. They use methane, which is alkane, as a carbon and energy source. And this one is actually aerobic process. Don't get confused. You have ANME, which is also methane oxidation, but it is anaerobic, and you have Candidatus methylmirabilis oxifera, which is also anaerobic, but it's also using methane. The difference is respiring, depending on the mode that methanotrophs assimilate this toxic formaldehyde, you have ramp cycle gamma methanotrophs and serine cycle alpha methanotrophs. Again, methanotrophs, I can't stress this enough. ANME, anoptic methane oxidation consortium with SRB. And these special types of metanotrophs that are called verrucomicrobia live at very, very low pH and a high temperature. They're specialized by assimilation of CO2. But one that I really insist is this NME plus SRB symbiosis. So again, this is why I'm stressing it out. If I ask you what are anaerobic methane degrading organisms, you will tell me I'm in collaboration with SRB, and you will tell me about NC10 phylum Candidatus methylomirabilis oxifera, and these special nitroreducents that are reducing NO3 minus, again, anaerobic respiration. Exception of them is this FJG1, which is called methylomonas dinitrificans, the one that is unique by producing nitrous oxide. The reason why I like it is because it was actually discovered in our university and lab where Brittany and I work. If nothing is available, metanotrophs will respire metals, iron and manganese. And last but not least, is Vinogradsky. I even can maybe finish. Oh yeah, I can. Vinogradsky column. There was a question, what you need to know about the Vinogradsky column. So this is what you need to know. This is what I am presenting now. The order of organisms from bottom to top. What goes on the bottom? You don't have to know how is Vinogradsky column made. You don't have to describe me. Take the bottle, fill it up with cellulose. No, you don't have to tell me about that. But you have to understand that in order to explain the order of microorganisms. So I will never ask you to describe how will you make Vinogradsky column, simply because you are not in first grade. So I will ask my son that when he grows up. And I will. I so will. So on the bottom will be cellulose degrading organism like Clostridium. So there will be fermentation happening because glucose will be fermented to organic acid and phosphodiet. Next one, like from the bottom to top. So I think so. This is where Clostridium will be. Cellulose originates from that newspaper that you will put in the column. Next one are sulfate-reducing bacteria, like the sulfovibrio, because it's requiring sulfate. Why that one is just above Clostridium? Because it will use organic carbon source made by glucose fermentation that was done by Clostridium. So the sulfovibrio will use those organic sources and respire sulfide, which means it will create sulfide. That means on the top of that will be bacteria that can actually consume this sulfide, purple and green sulfur bacteria, and it has to be closer to the top because it has to have light available. So they will use hydrogen sulfide made by this sulfovibrio, and they will fix carbon dioxide. On the top of them will be non-sulfur bacteria. Again, they will use right from the top. There is also organic that will be that will be made by Clostridium, but there will be no carbon dioxide fixation. They don't need it. They use organics. And on the top will be famous cyanobacteria because they're doing oxygenic photosynthesis. That's literally what I want you to know. And all life on Earth can be characterized as phototrophs and chemotrophs because they're obtaining energy from chemicals. They're chemotrophs. Chemolithotrophs if it's inorganic. Chemoorganotrophs if it's organic. Phototrophs if it's alive. Autotrophs if they fix carbon dioxide. Heterotrophs if they're using organic carbon. So that's all what I want you to know from these two lectures. And that would be all.